Okay. So dear students, in the previous class, we had studied about the cell cycle, isn't it? At present, we are going to discuss about the mitosis or equational cell division. Mitosis or equational cell division. So in the background slide, you can see that there are the cell division stages like late telophase. So early telophase stages you can find here. Almost the cells are in the process of dividing out. Okay, so that is the backdrop slide you have of cell division. So let us proceed with this topic. So mitosis is a cell division which occurs in the somatic cells or vegetative cells or non-reproductive cells. Okay, even in reproductive cells, they can undergo mitosis. We call it as haploid mitosis takes place. It is not that they won't take place, but majority of the cells where they occur is mitotic cell division. It occurs in non-reproductive cells, or we also call it as somatic cells, or in plants, you can call it as vegetative cells. Okay, so mitosis is also termed as equational cell division. So mitosis is termed as equational cell division, uh, where you can notice the number of chromosomes in the parent and the progeny cells, which we usually call it as daughter cells. It is going to be the same. See, if you're going to take the example of human beings, the diploid number is 46 chromosomes. So the 46 chromosomes parent cell, it undergoes mitosis to produce two daughter cells. Each daughter cell will again be having 46 chromosomes each. So the ploidy level doesn't change. The ploidy level remains the same in the parent as well as the offspring. So we call this sort of cell division as equational cell division. Mitosis is also called as equational cell division. So mitosis is generally noticed in diploid cells. Even in haploid cells, mitosis can take place. It is not that it cannot take place. It also occurs in haploid cells in some of the lower plants and some social insects also. You can notice that uh, haploid cells also undergo mitosis. Okay, it involves major reorganization of all the cellular components. In the mitosis division, you can see that it is a major reorganization of all the cell components. So here in the slide, if you can notice, this is the initial first stage is the prophase stage. So where there is a disintegration of nuclear membrane, nucleolus, they are all going to disintegrate. Okay. And you can also notice the movement of the centrioles towards the opposite side, which forms the asters. In the second stage, there is early metaphase it is. So you find that in the late metaphase, the chromosomes are all arranged on the equatorial plate and the asters are moved towards the opposite poles and they have the spindle fibers attached to the chromosome at both the end. So you know that the centromere of the chromosome, it has kinetochores. So the kinetochores are attached to the spindle fibers. Okay, from both the end. It is like just a tug of war. So in the next stage, you can notice that so the chromosome splits at the centromere because it is, as I told you, the spindle fibers from both the opposite poles. It is just like a tug of war. So the centromere, it breaks at the center in the mitotic cell division, and the chromosome with two arms, they move towards the opposite poles. So the chromosome splits at the center. So the chromosome which had four arms, it has split into chromosome with two arms. Okay, so or you can also say it as chromosomes with sister chromatids. They are split to form two sister chromatids. They are split to form a chromosome with single uh, sister chromatid at the centromere. So they have split at the centromere and they are pulled towards the opposite pole in the anaphase stage. So this is early anaphase and this is late anaphase stage. Okay. So after this anaphase stage, there is a reappearance of nuclear membrane, nucleolus, and this chromosomes becomes a chromatin material. Okay. And there is a beginning of division of cytoplasm. So you can see a furrow being formed by the cell. Okay, so then the cytokinesis also takes place and two new daughter cells are formed, which takes up the original size of the parent cell. So both the daughter cells will be having the same chromosomes as the parent cell. That is the reason this mitotic division is also called as equational cell division. So you can see the major reorganization of all the cell components during the cell division, and they come back to its original stage as that of the parent cell. So now two new progeny cells or daughter cells are formed from a single parent cell. Okay, so this is what is the summary of the entire mitotic cell division. So we'll be studying about it step by step. So the mitotic division, they have two stages, that is division of nucleus, which we call it as karyokinesis, and division of cytoplasm, which we call it as cytokinesis. So first we'll focus on karyokinesis. See, the karyokinesis of mitosis has four stages: prophase, metaphase. Metaphase is where the chromosomes are arranged on the equatorial plate. Then anaphase, where the chromosomes move towards the opposite poles, and telophase, so where there is a completion of karyokinesis. So telophase is followed by cytokinesis. So I've already told you in your regular practical class, what you would be doing is you'll be taking the onion root tips and you're going to just crush them, and you notice that the different stages will be there. Onion root tips you take because root tips have meristematic cell division they'll be undergoing. So you can easily observe the cell division stages. That is the reason we choose the root tip of onion. Okay, so you can notice the various stages, like it is a prophase stage, metaphase stage, almost the metaphase stage, late metaphase stage. Then you can notice this sort of crown like thing where it is very easy for you to differentiate the uh, anaphase stage. And almost here you can notice the furrow also being formed. This is a telophase stage. Okay, so telophase stage, there is a reappearance of nuclear membrane, nucleolus, and there is a furrow formation, which also leads to the cytokinesis. So the karyokinesis of mitosis has four stages: prophase, metaphase, anaphase, and telophase. But you should also remember, before this karyokinesis takes place, there is an interphase stage. So cell cycle, we have studied about that interphase stage, the gap one phase, S phase, gap two phase. So all those preparations leads to replication of DNA would have taken place. So there is double the copy of DNA, but same number of chromosomes, that you should understand. And there is also RNA and protein synthesized. All these are in expectation of a cell division to take place. So these preparatory phase would have happened in the interphase, which we are not discussing it here. But you should be aware that uh, each and every cell that enters through this karyokinesis would have undergone the cell cycle, wherein there was G1 or gap 1 phase, synthesis phase or S phase, then gap 2 phase. So where gap 1 and gap 2 phase, you know that there is a synthesis of RNA and proteins in both the stages. Whereas in S phase, synthesis phase, DNA is going to undergo replication, and they are going to form two copies of DNA, identical copies of DNA, but the number of chromosomes remain the same. That we have discussed in the previous class. So that stage is also there. So before karyokinesis, they would have undergone the cell cycle. After the gap 2 phase, they enter into the karyokinesis of mitosis, followed by cytokinesis in the M phase. This is the M phase, first step of it, that is karyokinesis of mitosis, which has four stages: prophase, metaphase, anaphase, and telophase. In the prophase, so it is the longest phase in mitosis. It follows the S and G2 phases of interphase. So there is synthesis and gap 2 phases of interphase. In the S and G2 phases, DNA molecules are intertwined. So the DNA molecules are all intertwined. You can notice this in the early prophase. So early prophase has a nuclear membrane, nucleolus, then the chromatin material. So you can notice the DNA molecules are intertwined in the prophase stage. So the characteristic events of prophase are the chromosome material or chromatin fibers are untangled and they condense to form mitotic chromosomes. Okay, the condensation process takes place of the chromatin fibers, and they condense to form the mitotic chromosomes. They are seen to be composed of two chromatids attached together at the centromere. So they have sister chromatids attached together at the centromere. You find the two sister chromatids, and they have four chromosomal arms. Okay, attached to the centromere, you find two sister chromatids. So two chromatids attached together at the centromere. So the centrosomes in case of animal cell divisions, not in plant cell division, in animal cell division, the centrosomes begin to move towards the opposite pole of the cell. Okay, each centrosomes radiate out microtubules called as, okay, they radiate microtubules called asters. The two asters together with spindle fibers, they form the mitotic apparatus. So the two asters, so the asters are nothing but each centrosomes radiate out microtubules, that we call it as asters. The two asters together with spindle fibers, they form the mitotic apparatus. So they might ask you, define mitotic apparatus. The two asters along with spindle fibers, they together form this mitotic apparatus. So mitotic apparatus is composed of the two asters along with spindle fibers, which we together call it as mitotic apparatus. So you have to remember that. So the chromosome materials, they are going to condense, the chromatin fibers are going to condense to form chromosome like structure. So the chromosome has sister has two chromatids attached to the centromere. That is, they have two sister chromatids attached to the centromere. Okay, so the centrosomes are going to move towards the opposite poles, and they are going to radiate out through microtubules called asters. So the centrosome along the two asters along with the spindle fibers, we together call it as mitotic apparatus. So cells at the end of prophase, so this is the late prophase, they do not show Golgi complex, endoplasmic reticulum, nucleolus, and nuclear envelope. They disappear or disintegrate. Cells at the end of prophase, they do not have endoplasmic reticulum, Golgi complex, nuclear membrane, and nucleolus. They're all going to disintegrate or disappear. So that is what you can notice in the uh, final prophase stage. So at the end of prophase, the cells do not have the nuclear membrane, nucleolus, Golgi complex, and endoplasmic reticulum. They are uh, disintegrated or they are disappear at the end of the prophase. That is late prophase stage. So the next step is metaphase. So what you can notice during metaphase is the arrangement of chromosomes at the equatorial plate, which we also call it as metaphase plate. So the center of this, see this center of it, we call it as the equatorial plate or metaphase plate. So you already know that at centromere, there are two sister chromatids, or you can also call it a centromere with four chromosomal arms. So two sister chromatids are attached at the centromere. So the nuclear envelope in the metaphase stage, it is completely disintegrates. So the chromosomes spread through the cytoplasm of the cell. So the chromosomes are going to float and spread within the cytoplasm of the cell. The chromosome condensation is completed. So the condensation of the chromatin fibers is completed in the chromosome. And I've already told you the best stage to study the structure of chromosome is metaphase stage. So the chromosome condensation is completed. So the chromatin material condensation is completed in the chromosome. So they can be studied under the microscope. They will have two sister chromatids attached to the centromere. So you can notice each chromosome will have two sister chromatids or four chromosomal arms. So each chromosome has two sister chromatids attached at the centromere. Okay. So chromosomes come to lie at the equator. So that is the important step that is taking place during the uh, metaphase stage. So they come to lie at the equator. The plane of alignment of the chromosome metaphase is called the metaphase plate. So the chromosomes come to lie at the equator. Okay. So the plane of alignment of the chromosomes at metaphase, it is called as metaphase plate. So the spindle fibers from both the poles, see, you can see the asters with radiating microtubules. So the centrosomes with radiating microtubules, we call it as asters, and the spindle arise from this microtubules and they are attached to the kinetochore of the centromere. Okay, so they have not described that, but you should understand that the spindle fibers are attached to the kinetochore of the centromere. So the centromere has kinetochores at both the ends here. So from the opposing ends, the spindle fibers are attached to the centromere. So we'll just take one example of this chromosome where you can notice that this chromosome centromere is attached with spindle fibers from both the ends. So I have already told you it is like a tug of war. So for when the spindle fibers condense, they pull them towards the opposite poles. So obviously the centromere will break at the center and the two sister chromatids, they break to form centromere with single chromatids and they move towards the opposite poles. So totally number of chromosomes are four chromosomes with two sister chromatids in each chromosome. But after this anaphase stage, what you notice is you'll be having four chromosomes at both the poles, but they'll have single sister chromatid because the centromere breaks at the center. Okay, so what are the things that you notice in this metaphase stages? The nuclear envelope completely disintegrates, the chromosomes spread through the cytoplasm of the cell. So chromosome, the chromatin material condensation is complete within the chromosomes. So the best stage to study the chromosomes are under this metaphase stage. Okay, so and you notice that the chromosome will have two sister chromatids uh, attached at the centromere. They'll have two sister chromatids uh, attached at the centromere. So that is what you have to remember. And chromosomes in the metaphase stage, they come to lie at the equator. The plane of alignment of the chromosomes at the metaphase. So this plane of arrangement of chromosomes at the metaphase plate, at the metaphase stage is called as the metaphase plate. So the spindle fibers from both the poles arising from the asters from both the poles, they are connected to chromatids by the kinetochores in the centromere. So you know that centromere will have kinetochores at both the ends. So the spindle fibers from both the poles are connected to chromatids by the kinetochores in the centromere. Okay, so at both the ends they are connected, and I told you that the centromere splits in the anaphase stage. Let us study about that. So the important phase, it is the shortest phase in the mitosis. So you should remember anaphase is the shortest phase in the mitosis, and the longest phase is prophase. So the shortest phase in the mitosis is anaphase. So the centromere of each chromosome, they divide longitudinally, resulting in the formation. See, there were two sister chromatids at the equatorial plate, but because of the condensation of the spindle fibers from the opposite poles, the centromere breaks at the center, and a single chromosome with two sister chromatids now, after the breakage at the centromere, they are going to form centromere with a single sister chromatid. So you can see that they have only two chromosomal arms. So they break at the divide longitudinally, resulting in the formation of two daughter chromatids, chromosomes of the future daughter nuclei. So totally the number of chromosomes has not varied, but the type of chromosome has varied. So you find four chromosomes at both the poles. Okay, but instead of them having two sister chromatids at the centromere, they have a single sister chromatid. Okay, so the centromere of each chromosome divides longitudinally, resulting in the formation of two daughter chromatids. So the chromosomes which are going to be the future chromosomes of the daughter nuclei. So as the spindle fibers condense or contract, the chromatids move away from the equator to the opposite poles. In the late phase, you can see that they have moved away from the, this is early anaphase where you can see they are almost at the equatorial plate or metaphase plate. Now they are condensation of the spindle fibers leads to them, the chromatids moving away from the equatorial plate towards the opposite poles. In the telophase, the final stage of mitosis, karyokinesis of mitosis, the chromosomes cluster at opposite poles. So the chromosomes are going to cluster at the opposite poles and they uncoil to form chromatin fibers. Okay, so the nuclear envelope reappears. So they assemble around the chromatin fibers. So thus two daughter nuclei are formed, and also nucleolus, Golgi complex, and endoplasmic reticulum, they reappear. The spindle fibers disappear. Okay, so these are things that are happening in the telophase. So the chromosomes they cluster at the opposite poles and they are going to uncoil to form chromatin fibers. So the nuclear envelope assembles around the chromatin network, chromatin fibers, and we call that as nucleus. So thus two daughter nuclei are formed at the opposite poles. So the nucleolus, Golgi complex, and endoplasmic reticulum, they reappear in the telophase stage. The spindle fibers disappear. Okay, so these are the changes that are taking place in the telophase. So the after this karyokinesis, the final stage was telophase of karyokinesis. The next stage is cytokinesis. Cytokinesis, as the name indicates, it is the division of cytoplasm. So there has been a division of nucleus to form two nuclei. Now there has to be division of cytoplasm to complete the mitotic cell division. So the division of cytoplasm results in the formation of two daughter cells. So you can notice here the formation of two daughter cells after the division of the cytoplasm. Okay, it starts. So the cytokinesis starts when telophase is in progress. You can see a furrow-like structure happening within the cell. So the cytokinesis starts from the telophase stage itself. So you can see a furrow-like structure happening. So here a cleavage furrow is appeared in plasma membrane. So the cleavage furrow appears in the plasma membrane. It gradually deepens this cleavage furrow and joins in the center, dividing the cytoplasm into two. Okay, in animal cell, what you notice is the cleavage furrow appears in the plasma membrane and it gradually deepens and joins at the center. See, initially it was only at the edges, the furrow, but it gradually deepens and joins at the center. From both the end, they are going to gradually deepen the furrow and join at the center. So, and now they have formed a plate or a membrane which separates the two daughter cells, dividing the cytoplasm completely into two. Okay, so the furrow gradually deepens and joins at the center, dividing the cytoplasm into two in case of animal cell. Whereas in plant cell, you find that there is a cytoplasmic plate formed at the center, which extends at both the ends, and they are going to form this cell wall. Okay, in plant cell, the cytokinesis in animal cells, it is different due to the presence of, see, in plant cell, the cytokinesis is different from animal cells due to the presence of cell wall. So animal cell did not have a cell wall, so it was easy. A furrow was formed with later deepened and joined at the center to divide the cytoplasm. Whereas in plant cell, they have a cell wall and it is difficult for them to undergo such sort of cytokinesis. So it is the cytokinesis division is different in case of plant cells. In plant cells, the vesicle formed from the Golgi complex accumulates at the equator. So the vesicles which are formed from the Golgi apparatus or Golgi bodies, they accumulate at the center. So the vesicles, okay, they are formed from the Golgi body, they accumulate at the equator. It grows outwards and meets the lateral walls. So the vesicle bodies, they grow outwards and meet the lateral walls. They fuse together to form a cell plate. So from the center, the vesicles extend outwards and they form a continuous wall, which we call it as the lateral wall. So they fuse together at both the opposite ends. So we call that as cell plate. So this cell plate separates the two daughter cells, the cytoplasm of these two daughter cells. Later, the cell plate becomes the middle lamella. So you should remember that in plant cells, the vesicles are formed from the Golgi bodies, accumulate at the equator. And these vesicles, they grow outwards. both the ends they grow outwards and meet the lateral walls. So they fuse together. So they meet the lateral walls and they fuse together to form a cell plate. This cell plate separates the two daughter cells cytoplasm. Later, the cell plate becomes the middle lamella. So in plant cell, the cytokinesis is different from that of animal cell because the plant cell has a cell wall. So there is no furrow formed which deepens and meets at the center. Rather, here the Golgi body produces the vesicles which are found at the equator. You can notice that the Golgi bodies have produced the vesicles which are found at the equator. They extend uh, outwards and meet the lateral walls. So they extend outwards both the sides and they meet the lateral walls. So they fuse. So these vesicles fuse to form a continuous plate, which we call it as the cell plate. And this cell plate separates the cytoplasm of the two daughter cells. Later, the cell plate develops to form the middle lamella. So the growing cell plate you can notice here, so which extends laterally outwards and it meets the lateral walls. Okay, it extends laterally outwards to meet the lateral walls and they form a continuous plate which we call it as cell plate. It separates the two daughter cells. So this is what is the cytokinesis, which is a different type of cytokinesis you can notice in plant cell. In animal cell, it was just the furrow that was formed during the telophase that deepens and meets at the center. That itself forms a cell wall, the cross wall, so that divides the cytoplasm into two and they form two daughter cells. That is in case of animal cell. So in plant cells, the Golgi bodies produce vesicles, and these vesicles they are going to extend outwards and meet the lateral walls. So they fuse to form the cell plate, which separates the two daughter cells. Later, the cell plate becomes the middle lamella. This is what is the process that is taking place in cytokinesis in plant cell during mitosis. Okay. So during cytokinesis, organelles like mitochondria and plastids get distributed between the daughter cells. So especially in plant cell, we are discussing about during cytokinesis, the organelles like cell organelles like mitochondria and plastids, they get distributed between the daughter cells. In some of the organisms, karyokinesis is not followed by cytokinesis. As a result, multinucleate condition arises. So that multinucleate condition arises because after the division of nucleus, there is no formation of cross wall. So we call such type of cell as syncytium. So the multinucleate condition is called as syncytium. So example is liquid endosperm in coconut is a very good example for the syncytium type of cell. So in some organism, karyokinesis is not immediately followed by the cytokinesis. As a result, multinucleate condition or syncytium arises. Example is liquid endosperm in coconut is a free nuclear endosperm, we call it as. So that liquid endosperm, the ಎಳ್ನೀರು, ನೀರು, the water of that, the coconut milk, what you call it as. Okay, so the fluid of coconut, so that tender coconut, that is nothing but liquid endosperm in coconut, and we call it as free nuclear endosperm because after the karyokinesis, there is no formation of cell walls. That is, there is no cytokinesis. As a result, multinucleate condition or syncytium arises. Example is liquid endosperm in coconut. So remember about that. So you can remember this overall steps of it. So in your book, they have given a very simple diagram. You can follow that. This is just for explanation part. So the mitosis, they would have undergone an interphase. Don't draw the same diagram. You can check the book and draw the diagrams that are in the book. So interphase stage. So the prophase stage. So where you can find the centrosomes moves towards the opposite poles and they have microtubules forms from the centrosomes, so which we call it as asters. So the asters microtubules will be producing the spindle fibers. So the two asters with the spindle fibers, we call it as the mitotic apparatus. So that you also have to remember about. This is a early metaphase, where the you can notice that the chromosomes are distributed in the cytoplasm. Later, they are all arranged at the equator. So we call this as the metaphase plate. So where you notice the chromosomes are arranged in the late metaphase. So the centromere of each chromosomes, the kinetochore of the both ends of the centromere, they are attached by the opposite spindle fibers from the asters. So then there is the early anaphase, where you can see that longitudinally the centromere splits. Okay, from two sister chromatids, the centromere will be now having single chromatid. Okay, and they move towards the opposite poles. So the opposite poles will have the same number of chromosome as that of the parent cell. So there is no difference in the chromosome number. That is why we call this overall mitosis is also called as equational cell division. So in the anaphase, which is a short phase, prophase is the longest phase of the cell division, mitosis. Then the anaphase in mitosis is the shortest phase. Okay, then there is the cytokinesis after the karyokinesis, where there is a reappearance of nuclear envelope, nucleolus, endoplasmic reticulum, Golgi apparatus. So there is a formation of cross walls, and they are going to separate to form two daughter cells. Okay, and both of them will have the same chromosome number as the parent cell. That is the reason we call this mitotic cell division as equational cell division. So that you have to remember about. So these are the overall steps of mitotic cell division. Now, what are the significance of mitosis? They produce diploid daughter cells with identical genome. So you should understand that they are producing diploid daughter cells with identical genome. So it helps to retain. Why is there an identical genome? Because during the synthesis phase, the DNA produces identical copies of DNA. That is the reason the genome will be identical. So it helps retain the same chromosome number in the somatic cells. So now when they undergo mitosis, any somatic cell, they'll have the same chromosome number. If they had haploid set of chromosomes, they'll have haploid set of chromosomes. If they have diploid set of chromosomes, they'll have diploid. So it helps in the body growth. See, mitosis is, see, whenever I talk about meristematic cell division, it is mitosis. So it helps in the body growth of multicellular organisms. Okay, mitosis in the meristematic tissue, they are going to help in a continuous growth of plants throughout life. Okay, it restores the nucleocytoplasmic ratio. See, the mitotic cell division, one of the other important pointers, it restores the nucleocytoplasmic ratio that is disturbed due to cell growth. So it always tries to restore the nucleocytoplasmic ratio. Whenever there is a difference in nucleocytoplasmic ratio, they undergo cell division. Okay, so mitosis tries to restore that nucleocytoplasmic ratio that disturbed due to that was changed due to the cell growth. And it also helps in this mitosis also helps in cell repair and replacement. I told you the healing process in us whenever there is a injury or wound or cut of skin, it is by mitotic process. So it helps, and mitosis helps in cell repair and replacement. Example, cells of the upper layer of the epidermis, lining of the gut, and blood cells. So the dead and worn out cells are replaced by new cells by the process of mitotic cell division. So mitosis helps in cell repair and replacement. Example, cells of the upper layer of the epidermis, lining of the gut, and blood cells. They are all replaced by this process of mitosis. Okay, so one what I used to do was just to drive home the point importance of meristematic tissue as a growing point. So after the students had left the onion in the lab where they have cut the root tips, I used to send the onion back to the students and ask them to grow it. So what do you expect the result to be? So all of them who had cut the onion root tips properly, they came and reported to me, sir, there is no growth of the onion and the onion has decayed. Why is it so? Why did the onion decay after cutting the root tips? See, they did not have any more of meristematic tissue. That is the reason when I gave them the onion without the root tips, they had all cut the root tips for the mitotic cell division. So when I gave them such onions, they were not able to grow because the meristematic tip, which is found at the root apex, they were all completely lost. As a result, the onions decayed. They did not have a growing point. Okay, so it helps. The mitotic cell division helps in the body growth of multicellular organism. Mitosis in the meristematic tissue helps in the continuous growth of plant throughout the life. So the meristem, you know, that apical meristem, lateral meristem, intercalary meristem, they are responsible for primary and secondary growth. So the mitosis in the meristematic tissue helps in the continuous growth of plants throughout the life. Okay, so whenever the nucleocytoplasmic ratio is disturbed due to cell growth, this mitosis restores it. By the mitotic cell division, the nucleocytoplasmic ratio is restored. Okay, so these are the various significance of mitosis or importance of mitosis. So you have to write all this. It produces diploid daughter cells with identical genome. It helps to retain the same chromosome number in all the somatic cells. It maintains constant ploidy. Okay, so it helps to retain the same chromosome number in all somatic cells. I've already told you why they have diploid daughter cells with identical genome, because DNA duplication or replication produces identical copies, and there is no recombination here as it happens in meiosis. There is no gene recombination. As a result of it, the diploid daughter cells will have identical genome as that of the parental cell. So what is genome? The sum total of haploid set of chromosomes is called as genome. See, the sum total of genes present in haploid set of chromosomes, we call it as genome. So definition of genome is the total number of genes, the sum total of genes present in haploid set of chromosomes, we call it as genome. So you should remember about that. So the genome of the daughter cells, it is not going to be different from that of the parent cells, and they are identical because the DNA replication also is an identical copies it produces. Semi-conservative mode of replication I told you about, which we will discuss in the uh, next academic year. Okay, so have you understood about this topic? You have any doubts or clarification, students? Any doubts or clarification during mitosis?